So in this series of videos, we're going to cover specific immunity and extend from our lecture, from the last lecture about non-specific immunity, which was those first and second lines of defense in the immune system. So remember that the first line of defense was really just some chemical, genetic, and physical kind of barriers. And the second line of defense was all of those non-specific things. So things that happen where the cells that are responding aren't responding because they are recognizing a pathogen. They are responding because they just recognize that something is foreign in the body and it's not you. And the four different parts of that second line of defense were inflammation, interferon, complement, fixation, and phagocytosis. So when we move into the third line of defense, this is going to be where there is specificity. So actually the cells are recognizing that pathogen and they are responding specifically to that, and we'll talk about how that happens. And as a result of responding to a pathogen, they actually remember. The body has some sort of memory, so if we are ever exposed to that pathogen again, there is a stronger and more efficient response. So this is the type of immunity that you build up over time, so that you can we can fight infections, by exposure. And this is the kind of immunity that you develop as a result of a vaccine and being exposed to something that is similar to a pathogen. So this slide right here gives you a big picture and overview of everything that's going to happen. So I'm going to start by walking through all of this, and then I kind of cut back and we'll go into a little bit more detail about things. So the cells that are really going to be predominantly involved in this are B cells and T cells. And I actually do have a video already posted on Moodle that just contrasts B cells and T cells in general. And so we'll look at where they come from, how they're made, how they're specific, and then if they are activated and actually, are involved in the immune response, what is that mode of action and how do they develop memory? So, again, we could come back to this at the end and make sure that we understand all of the pieces in there. So let's just look at an overview of what happens. We looked at hematopoiesis of human blood cells. And we talked about some of them like neutrophils and macrophages and dendritic cells a little bit. We'll talk about them again too here. But we had mentioned B cells and T cells, but now we're going to talk about them more in detail because they are a part of the specific immune response. So remember, all of the differentiation of all of the different types of blood cells happens within the bone marrow for adults. So B cells and T cells, they are both going to differentiate or be formed within the bone marrow, but then they migrate to different locations within the body where they actually undergo a process of maturation, which I'm going to talk about. So B cells actually go to specialized sites within the bone marrow to mature, whereas T cells, they migrate to the thymus in order to mature. Once they're done maturing, then both the B cells and the T cells end up in your lymph nodes and in lymphoid organs, and that's where they sort of just hang out and wait to have a job. So what happens then is essentially if you're exposed to some sort of foreign cell, all of those first and second line of defense things are going to happen, right? So the foreign cell has to get across that first line of defense, past those physical, chemical, and genetic barriers, and then, get into the tissues, right? And then when that starts to happen, we immediately see that process of inflammation and phagocytosis, where we're pulling fluids into the sites and macrophages into the sites, and we're eating up like all of these foreign cells, trying to get rid of them, right? And then we also have dendritic cells and neutrophils and macrophages that act as what were called antigen processors and presenters. So as these cells are sort of engulfing and breaking up things, they actually take little pieces of these weird foreign cells, these pathogens that have come in, and these little pieces are called antigen, and they are able to then go off to the lymph nodes and say, hey, is this something that one of the B cells and T cells would respond to? And if so, can you come back and help out? Right? So there is some distinction. That's sort of an oversimplification. Like I said, I'll go back and talk a little bit more about in detail about this process. But somehow, some part of that foreign cell is going to what we call activate the T cells and the B cells so that they can, perform their function, right? Once the B cells and T cells are activated, so they recognize this antigen and the specific, what we call clone, one of the specific B cells and T cells, says, oh, hey, I recognize that antigen, I can work on it, and so they start to proliferate, and they start to make copies or clones of themselves, and they're all genetically identical, so whatever T cell makes a clone of itself or B cell makes a clone of itself will also respond to that antigen that's present. And some of those cells, the clones, they go fight the battle, so they're the troops, or they're involved in fighting the infection. And then some of the cells actually just stick around in the form of memory, right? The clones of B cells are actually called plasma cells. So plasma cells are those ones that go out and fight the infection. And B cells, actually, sorry, I misspoke. But plasma cells don't really go out and fight the infection. This is one of the differences between B cells and T cells. B cells actually just stay in the lymphoid tissue, and they are exposed to antigen, and as a result of it, the clones actually produce something called antibody, and they secrete that antibody out, and it goes into the tissues and into the blood, so the antibodies are actually fighting the infection. When the antibodies attach to antigen, they sort of mark whatever that cell is for destruction, right? So it comes into play when we talk about complement fixation and like alerting macrophages. And there is a video that I have that talks about all of the four functions of an antibody just in and of itself, but also it will be incorporated into the end of this video or next one if I run out of time. So the function of antibodies as a result of the B cell clones and what the plasma cells are doing is called humoral immunity. And just keep in mind, remember that the B cells stay in the lymph nodes. They don't go out and fight the infection. They actually just pump antibody when they are activated. So what do the T cells do? So when the T cells, they recognize antigen, and when they become activated, the T cells start to clone themselves, right? But there's two different kinds, and this is a little bit of a simplification, but there are cytotoxic T cells and there's helper T cells. These cells actually leave the lymph nodes, unlike the B cells, and they go and they fight the infection, right? Cytotoxic T cells, they actually go and kill host cells and foreign cells. So why would you want to kill a host cell? Well, if a host cell was infected with a virus, you would want to kill it off so that it wouldn't be replicating the virus inside, or it would be able to kill the foreign bacterial virus, right? Helper cells, they help to activate other T cells and to communicate with macrophages and B cells. Remember that we've said that there's a lot of like intercommunication through cytokines, chemokines going on between all sorts of immune cells. So in a later slide, I'll actually show some specific details or an example of what a T helper cell might do. So keep in mind the specificity is all based on the major histocompatibility complex. The major histocompatibility complex is your cell receptor, it's your self marker. So it is an immune cell, or it is the signal to all immune cells that whatever this cell is actually you. So your immune system will not respond to or attack a U-marked cell. Now, I mentioned in the last lecture, too, in class that, autoimmune diseases and transplant problems, this is because of the major histocompatibility complex. An autoimmune disease is when your immune system starts attacking your own cells. So this is when this process or this complex has malfunctioned such that the immune cells are not recognizing your own major histocompatibility complex and they're starting to damage your own cells, thinking they're foreign. Transplant problems arise when you put, another person's kidney into your body. The major histocompatibility complex or self-marker on that kidney is different, so the immune system is going to recognize it as something foreign and try and attack it. So your entire life you take immunosuppressants in order to, keep your body from fighting off or rejecting the organ. All right, so B cells and T cells have receptors on their surface that recognize antigen. When, again, antigen is just any sort of foreign thing that will elicit a response or activate a lymphocyte, a B cell or a T cell. So B cells have receptors that bind directly to antigen. So antigen can just drain from wherever the infection is. It can go into the lymphatic system and drain up towards the lymph nodes, and it just rushes in the lymph node area, and it will bind onto a B cell, and the B cell can start making antibody that it'll pump out and send to the site of the infection. The T cells, however, their process is a little bit more complicated, and in order for their receptor to bind to antigen, it also has to bind simultaneously to major histocompatibility complex. So T cells can't just react to antigen that's around, they have to be presented antigen by an immune cell that has major histocompatibility complex. So the immune cells that can do this are like dendritic cells and macrophages. So they would take a little bit, or even other T helper cells, right? So they take a little, B cells can do this. So they can take a little bit of antigen and they can, show it to the T cell, and the T cell can recognize and grab onto the major histocompatibility complex of that dendritic cell or that host immune cell, and then those T cells get activated. All right, I'm running a little short on time, so we're going to leave it at that, and then I'll come back and actually talk about the genetics of these receptors and how we have genetic diversity such that the receptors can respond to antigen that comes from the outside environment.